Hello everyone, welcome back to AS and A-Level Biology with Dr. Demi. I am Dr. Demi, and in today's video I am going into details of respiration. I'm going to be discussing all the steps that are involved in respiration, which include glycolysis, the link reaction, the Krebs cycle, and oxidative phosphorylation, which is also called the electron transport chain. And I hope that you find these videos really helpful. Again, for those of you who might have just found this channel, I am posting the content in chronological order, so it is easy for you to follow it. As long as you know what chapter you're trying to study, you can simply just say, okay, I'm looking for chapter 12, and if you see chapter 12, you'll see about 4 videos that have broken the chapter into different sections so that they're easy for you to follow. So yes, let's get into it today. Let's look at respiration in detail. So first things first, when we say what is the main source of fuel for cells, the main source of fuels, of energy rather, or fuel, whatever you want to call it, is glucose. The body typically prefers to use glucose whenever we need energy, and that is why they say you exhaust your glycogen reserves first when you go on to exercise before you start to use up your fat reserves. The body can also use fat for energy whenever glucose is not present, and these are some of the things that form the backbone of famous diets like the keto diet. The fact that if you don't have glucose available, but you've eaten a good amount of fat and proteins, just by the way, that's an amino acid, then you would be able to supply your body with energy. But your cells would typically always prefer to use glucose. Now, respiration is a 4-step process, and here I have just broken it down so that you know what step is happening when, and I am going to go in for each section just in detail so that you understand it. I promise you it is not difficult at all to know. Just take the keynotes down so that you're able to follow. So again, that's why I said at the beginning, or I don't know if I said it, I can't remember, but if I didn't, please grab a notebook and a pen so that you're able to just take down notes of what is really important. So the very first step, like I showed you on the last slide, is glycolysis. And the glycolysis can happen either under aerobic conditions. When we say it's aerobic, it means that oxygen is present, or it can happen under anaerobic conditions, which means oxygen is absent. For the sake of this video, I'm only going to focus on aerobic conditions, and in the next video, I will then focus on anaerobic conditions because I just don't want things to get mixed up when you're learning. So we start off with glycolysis, just like the name suggests. I'm just going to annotate here on the slide. Just like this name suggests, glyco, that refers to glucose, and you can just call this lysis, which then tells you this is the breakdown of glucose. So glycolysis in simple terms is splitting glucose, which is a 6-carbon sugar, because the formula for glucose is C6H12O6. So we split the 6 carbon. So when we count the number of carbons, we typically refer to compounds by the number of carbons that they have when we are discussing respiration. So we split the 6-carbon compound into 2 molecules of a 3-carbon compound called pyruvate. And pyruvate is the one that is then carried on into the next steps of respiration. But before we start jumping into the next steps, I want you guys to be able to understand the different steps of glycolysis. I have summarized them here, sort of. I'm just telling you that glucose is phosphorylated and then converted to fructose and and that might sound a bit difficult. So on the next slide, I've actually outlined the steps in a diagram. So here we are. This is glucose, which is what we start off with. So when you go home, for example, after a long day, and you make yourself a very lovely sandwich and you eat that, you've taken glucose into your body, and perhaps you still need some energy for work, and the glucose is not being stored because you're really energy deprived, so your body's just trying to make more energy. The first thing that happens is that ATP will be used. And please don't be worried about this, because students typically say that if respiration is about producing ATP. Why are we using ATP? Well, think about it in terms of money as well. This is a famous saying. I don't know if it's true, but people say you need to use money to make money. So in this case, we need to use ATP to make ATP. So we use ATP here, and what we are basically doing is we take a phosphate from ATP and we add it to glucose. So that then produces ADP and phosphate, as I showed you in the previous video, that when you hydrolyze ATP, you're going to make ADP plus phosphate, and it's a reversible reaction depending on what condition you're under. So we take a phosphate from ATP and we add it to glucose. That then makes it glucose phosphate. The reason why there's a 6 here is that that ATP, or rather that phosphate, is added to the carbon, the 6th carbon on glucose. So if I can just draw quickly here what glucose looks like. Glucose typically looks something like that. It has a hexagon shape. I'm not the best at drawing, so if these start to look a bit weird, just don't laugh too much. So here we go. So there, and over here, this is our CH2OH. And in case you're confused on how to count the carbons in glucose, know that we always start from here. So the edges are the carbons. So this is 1 carbon, that's carbon number 2. This is carbon number 3. Here it is carbon number 4. This over here is carbon number 5. And this is carbon number 6. Okay, so again, you can watch the chapter 2 videos in order to get more understanding on this. But we add the phosphate here to the 6th carbon. Once we've done that, we then have this enzyme called phosphoglucose isomerase. And just like the name suggests, it is an isomerase. So it converts glucose into its isomer, which is fructose. It converts glucose to fructose, and then that becomes fructose 6-phosphate. We then use ATP again to phosphorylate fructose 6-phosphate, and this time we add the phosphate to carbon number 1, which is this one over here that I'm just circling, and so we end up with fructose 1,6-bisphosphate. The reason why it's called bisphosphate is simply because there are 2 phosphates that have now been attached to fructose, and then it would undergo different reactions. For your syllabus, luckily, you don't have to know the intermediate reactions. You simply need to remember that fructose is then converted to triose phosphate, which are 2 molecules, and these are both 3-carbon compounds over here. They're both 3-carbon compounds, and when it's converted over here, that doesn't look like a 3. I'm just going to erase it and write it again. So this is also a 3-carbon compound here. So when triose phosphate has been formed, it is then dehydrogenated. So a hydrogen is removed, which is what this is referring to over here. The hydrogen is removed and it is added to a compound or a coenzyme called NAD. NAD is then reduced. So remember, if you do chemistry, the addition of hydrogen to a compound is called reduction, and the addition of oxygen to a compound is called oxidation. So in this case, NAD is then, it becomes NADH because hydrogen is removed and added to it. And then triose phosphate then undergoes what's called dephosphorylation. In other words, phosphate groups are removed from triose phosphate and added to ADP in order to make ATP. And the result of that, once all of these reactions have been completed, is that we then are left with 2 molecules of pyruvate, which are 3-carbon compounds. So just to wrap up this and to sort of help you understand it better, always remember you start off with glucose, you phosphorylate glucose, which means you use ATP. Phosphorylation is the addition of a phosphate group. You use ATP and it becomes glucose phosphate. It is then converted by an enzyme to fructose phosphate. Fructose phosphate is phosphorylated again and it becomes fructose biphosphate, and fructose biphosphate is then split into 2 molecules of triose phosphate, which are 3-carbon compounds, because remember fructose is also a 6-carbon compound. So we then have 3-carbon compounds. These 3-carbon compounds, which are triose phosphate, are then dehydrogenated in order to form NADH. NADH is important in the production of ATP. So we have NADH that has been formed, and this is dehydrogenation, which is the removal of hydrogen. And then we have the removal of phosphates from triose phosphate, which means it is dephosphorylated. And so here we then have two phosphate groups removed from here, and we are then making ATP through a series of reactions, obviously, that you don't have to know. And then we have pyruvate at the end of glycolysis. So the end of glycolysis under aerobic conditions will yield pyruvate. At the end of glycolysis, we also yield 4 ATP molecules. But because we've used 2 ATP molecules at the beginning when we were phosphorylating glucose and fructose phosphate, it means the net gain of ATP in glycolysis is 2. We also make 2 molecules of NADH. But at the end, I have a table that shows you how this sums up really nicely. Once glycolysis has been completed, the cell then undergoes what we call the link reaction. The link reaction is simply where pyruvate is prepared for entry into the Krebs cycle. So what happens here is that pyruvate is first of all decarboxylated. Decarboxylation means the removal of CO2 over here. So once we decarboxylate pyruvate, remember that it is a 3-carbon compound. So when you remove a carbon dioxide from it, you've basically removed one of the carbons, and so you end up with a 2-carbon compound called acetyl. Over here, you also have NAD, and pyruvate is dehydrogenated, which means a hydrogen is removed from pyruvate and added to NAD to make NADH. Again, NADH is really, really important when we are making ATP in the final stage of respiration. The last thing that I haven't included here, and I'm just going to draw it, is that we then have the addition of coenzyme A, which I'm just going to write here as CoA. Coenzyme A is then added to this product, which is acetyl over here. Acetyl is the 2-carbon compound you form after you decarboxylate and dehydrogenate pyruvate. And when you combine it with CoA or coenzyme A, it is used to carry acetyl into the Krebs cycle, which I'll show you on the next slide. So just for fun, like a fun fact, coenzyme A is made up of a nucleoside. It has adenine and ribose, and it also has a vitamin, and its function is to carry acetyl groups to the Krebs cycle. So this here is the Krebs cycle, the much dreaded Krebs cycle, but there's no reason to dread it because, again, for your syllabus, you're not required to know many of the intermediates. What you are expected to know, however, are the compounds, the key compounds that are formed, and how many NAD, ATP, and FAD you make from this process. So here again is the link reaction. As you can see here, it is depicted in this image I got from the textbook. And so pyruvate is decarboxylated, dehydrogenated, and the product is added to coenzyme A. When the acetyl travels into the Krebs cycle, so I'm just going to use a different color here because they've used red, and I don't want you to be confused. So when the acetyl travels into the Krebs cycle, notice what happens. Coenzyme A does not go into the Krebs cycle with it. It delivers acetyl into the Krebs cycle, and then it is recycled to go and bind to the next acetyl that has been formed from pyruvate. Now, this acetyl, which is a 2-carbon compound, comes into the Krebs cycle, and it binds with oxaloacetate over here. Take note of this compound because when we discuss photosynthesis, you will encounter it again. It binds with oxaloacetate, which is a 4-carbon compound, and oxaloacetate plus acetyl then form a 6-carbon compound called citrate. And then that goes through different processes, and you have a series of decarboxylation reactions. As you can see here, CO2 is released, CO2 is also released over here, and you start to see that it continues to break down, and you also make NADH, so that's what we call reduced NAD, which means there are also dehydrogenation reactions happening. You also have the synthesis of ATP, you have the making of FAD as well, and more NAD. This is what you have to know. You don't have to know all the compounds that are in the midst of all of this, because your syllabus doesn't require you to do that. So basically the process is that citrate, the 6-carbon compound, is decarboxylated, it is dehydrogenated, it yields CO2, it yields hydrogen ions. Those hydrogen ions are added to NAD, and then you make NADH and FADH. NADH and FADH are simply electron carriers. So always remember that. Coenzyme A delivers acetyl to the Krebs cycle. It doesn't go into the Krebs cycle itself. FADH and NADH carry electrons, which are very important for the synthesis of ATP in the final stage. For every turn of the Krebs cycle, you make 1 FADH, 3 NADH, and 2 molecules of CO2 are given off, and you also make ATP. And I see here that I have just written FAD. Please take note that this is reduced FAD and not just FAD or NAD. So reduced NAD, reduced FAD as well. Now we come to the last part of photosynthesis, where things get really interesting. All of the products that have been made from the earlier stages, and the key products are NADH and FADH, are then brought into the electron transport chain. And I'm going to show you, by the way, where each stage of photosynthesis happens. So in case you're wondering about that, don't stress too much. So the FADH is brought here, so is the NADH. And what you can see here, it starts here, it says the reduced NAD. Over there, and the hydrogen is basically removed from NADH, and then it's turned into NAD. That hydrogen, remember, hydrogen is an atom, and when you split an atom, you're going to have an electron, okay, and you're also going to have a proton, which is basically hydrogen ions. The electrons that are made, oh, that was a bad drawing. The electrons that are made here from the splitting of hydrogen, as you can see, are transported. If you follow these red arrows, look at that. They are transported through a series of electron transport carriers, or electron carriers. And as the electrons move, they release energy, because electrons are known to be high energy molecules. So as they move from one electron carrier to the other, they release energy. And the terminal point for these electrons is oxygen. Oxygen is their final electron acceptor, and I'll explain how this then comes into play. The proton that is released from the splitting of hydrogen is pumped into the intermembrane space. Now, this is important, and I think here I need to really explain to you that respiration happens in the mitochondria. So I'm just going to do a drawing of what might be considered the mitochondria here. The mitochondria, if you remember from chapter 1, is a double membrane organelle. This is its cytoplasm. This folded part here is the inner membrane, and this here is the outer membrane. All right. So the protons that are made from splitting of hydrogen are pumped into the intermembrane space. So they're pumped here, where I'm doing the blue dots. They're pumped there, and they start to increase in concentration over there. Now, because the cells want things to be in balance, they're not trying to have a lot of protons on the outside, because then that makes the intermembrane space more acidic, because those are the things that protons contribute. The protons are then brought back into the matrix of the mitochondria. We call the matrix of the mitochondria its cytoplasm. That's also what the cytoplasm is, but the technical term is to say it's the matrix. The protons are pumped out, and then they have to come back in. Now, because they are charged molecules, they can't come in through the phospholipid bilayer of the cell membrane. They have to come in through a proton carrier. So they come in through this proton carrier, which also happens to be ATP synthase. Now, from the name ATP synthase, you can already guess this enzyme or this carrier synthesizes ATP. So the protons, as they come in, the electrical energy that they carry is used to power ATP synthase into coupling ADP with phosphate in order to make ATP. And I know for some of you that might just be like, oh my goodness, what's happening? But I will explain this again. So the protons then travel down through the carrier and they come here where they meet back with the electrons that they were once coupled with, and they are then added to oxygen, and that is how we make water. And that, of course, is how we have come about the formula for respiration, which is, if we were using glucose, C6H12O6 plus oxygen, and that will then give us 6 CO2 plus water. This is the reaction for respiration based on this. And remember, we made our CO2 during the Krebs cycle, and we make water during the electron transport chain. If this has flown over your head, please don't be worried. I'm going to explain it again on the next slide. And oh, just to say that this step yields 28 ATP, but I'll show you that again at the end. All right, so now let's look at this again. So we have the oxidative phosphorylation, and it is said to take place in the inner membrane of the mitochondria. As you can see from this image from the textbook, this is the intermembrane space, which tells you this is the inner membrane and there is an outer membrane somewhere here. Okay, so we remove hydrogen, and I've outlined the steps over here. We remove hydrogen from NADH and FADH over here. All right, and then we add that hydrogen into what's it called? We split that hydrogen, we split that hydrogen into protons, which is positive ions, and electrons. Okay, the protons are pumped out of they're transported out to through proteins into the intermembrane space, as you can see over here. And the electrons are transported through electron carriers until they find the final electron acceptor, which is oxygen. That is why we use oxygen in aerobic reactions or aerobic respiration. As for the protons, they start to accumulate on the outside over here. And that means that their concentration between the outside or between the intermembrane space and the inside, which is the matrix of the mitochondria, are not matching. And so what they're going to do in that case is they have to come back to balance things out. And so the protons then move through this proton carrier here, which also happens to be ATP synthase. As the protons move into ATP synthase, the energy, right, the electrical energy that they carry is able to power ATP synthase into coupling ADP and phosphate in order to make ATP. And the protons themselves will travel down here, combine with electrons, bind with oxygen, and then form water. For those of you who might be wondering, where does the ADP and the phosphate come from? Always know that the ADP and phosphate are present already within the cells, within the mitochondria. However, they need to be synthesized through very specific steps. So if ADP and phosphate are just hanging around, they are useless until they are coupled together to make ATP. And so that is what happens here. And this process is often called chemiosmosis. I hope that was clear. If it wasn't, I really hope that you reach out in the comments and just ask a question, and I will be happy to answer it. So here I have included this just to show you where the different steps of glycolysis happen. Steps of respiration. So if you look here, you will see that this is the cytoplasm. I'm just going to highlight the important parts. This here is the cytoplasm. This very lovely shape over here is the entire mitochondria. That tells you that this is just inside the cell cytoplasm itself, while there are other things that happen within the mitochondria. So first things first, glycolysis happens in the cytoplasm, as you can see over here, and that's where you form pyruvate. Pyruvate then goes into the mitochondria, where it undergoes the link reaction in the matrix of the mitochondria. The matrix of the mitochondria is basically the cytoplasm of the mitochondria, but it is not called the cytoplasm. It's often referred to as the matrix. So that's where the link reaction happens. The Krebs cycle also happens in the matrix of the mitochondria, as you can see over here. And you then have oxidative phosphorylation, which happens, as you can see here. This is the cristae. which happens in the inner membrane. That's why you have this bulge over here. It happens in the inner membrane of the mitochondria. So these are just where the different reactions occur. But if you're not clear, I'll try as much as possible to post. I will post it, not try. I'll post it in the description so that you're able to see. And so just to close things off, because I'm sure this was a bit of a heavy chapter, how much ATP do we make at every stage, which is what is really important. And this is usually a question that you might get asked in the exam. So for glycolysis, for example, we make 2 ATP, and we use 2 ATP, take note of the headings, and we make 4, which means that we have a net gain of 2 ATP. For the link reaction, we don't make any ATP at all. The Krebs cycle, for all its complication and the headache that it gives many students, only makes 2 ATP. So we have a net gain of 2 because we don't use any ATP in the Krebs cycle. And oxidative phosphorylation, which is the last stage that involves the transport of electrons, we make 28 ATP there because the electrons carry so much energy, and the movement of the protons power ATP synthase to make lots of ATP. And so at the end of that, we make 28 ATP. We don't use any ATP. And so the total amount of ATP that we would make from aerobic respiration is 32. So that is our final amount of ATP. I'm going to stop here now. I'm sure this is a little bit heavy. Try to watch it again if you feel like you missed a couple of things. And if you have any questions, just post them in the comments, and I promise to get back to you as soon as possible. Thank you for watching, and I hope that you're able to interact with the rest of the content even better. And yes, have fun, have a good time. Until the next video, goodbye.